Okay, today we are going to talk about the probability and distributions. Let's see, we will first jump into some kind of normalization problem. When you see, okay, I will normalize my matrix table and what you are doing. And then we will move to the concept of this generating model. You may heard about the people say, okay, I model my data using some kind of distribution. I would like you to know at the end of the lecture, and you know exactly what they are talking about, why they are doing the modeling, and the purpose of that, how they actually doing that. And then we will mention some commonly used distributions. We will talk about normal distribution, Poisson, and the negative binomial, and T distribution, because we are going to use this concept in, for example, the t-SNE plot. You have to know, okay, what's the difference between normal distribution and the T distribution, and then you will appreciate why people want to use T distribution to generate their data points at the end. Then we will mention the descriptive statistics. Just very quickly, just go through those, and mean and variance, coefficient of variance, etc., etc. And then we will see how can we calculate covariance matrix and what that means as well. At the end, we will mention the KL divergence. It's very, very commonly used when people try to fit one model with another and see the difference between these two. And, okay. So here is UMI matrix. So I'm going to talk about this normalization. People say, okay, I want to normalize my UMI count matrix by liberal size. After explaining this, I will use chalkboard to explain the data point and the feature. And sometimes you transpose your matrix. And then when you compute variance and covariance, sometimes you are not calculate for the data point, but calculate for the variables, that feature. So you have to make sure you know what I'm doing exactly. And I will take time to explain the cosine distance again, and how the cosine distance correlated with the covariance. Okay. Now, look at this table. That's a real data, and you have rows are the genes, and the columns are the cells. So you can think each column is a data point, which is a cell, because on your mind, you consider the data point of the cells. Or we can call it the samples. In the traditional way to do the RNA-seq, you are not sequence each individual cell, you sequence by samples. You have sample 1, 2, 3. In here, we have just thousands of samples. Each sample is a cell, so that's the difference. And the genes are the variables to define your state of each cell. So we should consider genes as variables to describe each of those. In statistics, people normally transpose this. You have a sample in the row, and you have a column are the features or variables to define your samples. Okay. In UMI matrix, we just transpose it. But you have to be very clear what you are doing. If I don't tell you what row is and what the column is, will you be able to recognize those? If I mask had this, the rows and the column, the table, will you be able to look at the matrix and say, okay, I'm looking at a matrix, which columns are the cells, rows are the genes. It's possible, right? You should be able to see. You see this 1, 2, 3, 4. The fourth gene, you have all cells have a similar level, but a really high. There's a kind of housekeeping gene. Expression level is high, but when you look at the column, you see the different kind of a distribution. Okay, so you're doing some kind of visual detection of a distribution. You can actually look at this and then figure out, okay, the columns are the cells, the rows are the genes, right? Okay. We will come back to this concept later. So now, if I, for illustration purpose, I just make a simple illustrative sample here, but exactly the same meaning because that's the same, the real data, but this is a toy data. In this case, a little bit hard to recognize which is which, because like a little bit random, okay, in here. But still, you have a lot of zeros because in real data, you do have a lot of sparse position. Okay, so assume we have columns of the cells and the rows are the genes. And the first question is, do we need to do the library size normalization? So library size is a conversion with name come from a traditional RNA-seq. It's not a good name, but people still use it. Use the library size as a number of the rates in each cell. Here, race is not a race, it's a UMI. You know, you have more race than UMIs because UMIs are unique identifiers. If I for the given number of cells, if I sum the rows, I get a number something like this. The first gene have a 16 in total, and the second gene have 9. So I have a mean 5.2. I can do this. I can do column, right? Assume you. So what kind of factors can have impact on this summaries? So why this gene have a 10 total number of rates or UMIs, and this gene only have 2. This cell. So there are multiple factors will influence this number, right? So what can you name some of them?
Different cell types.
Different cell types, yes. So some cell may be biologically larger than another. So you have a bulk of mRNA concentration, and also the total amount is larger than that. And also another factor will be the sequencing bias. By chance, it's not. So we want to actually control for the sequencing bias. The sequencing bias will overwhelm the biological variability. So the library size normalization is trying to get rid of those sequencing bias, and you have a different number of the total rates in each cell. What we can do is to sum. So we get a 7 and 5, and we normalize, and at the end you have the average for all the columns. The sum of the columns are the same. Okay, we normalize by the size of the total number of the rays in each cell. That total number of rays have the name called the library size. So if it's 10 is too larger than average, you just divide by a factor. If it's too small, you just multiply by a larger factor. At the end, you make the library size, the sum of the library sizes equal across all the cells. Okay.
Normalize to your string relative expression of each entire cell.
Yeah. So look at this. Remember, this is a really small, and we make this expression larger, and we just assume because of detection and bias, we having the sequence it into a sufficient level to express, to capture its real expression. So it's not perfect, but this is normally we can eliminate this so-called library size difference. Okay, it's commonly used by combination. There are certainly better ways to do it, but we will touch on it later. You have to understand your distribution, and then you have a better way to do it. But this is a very, very efficient computation. So take no time is click button, get this. Other method, you have to model this distribution and assume the zero is not zero. You have impute the way to do the imputation. But for the interesting of the balance between the computational efficiency and the sophisticated model, you have to choose. So library size normalization normally is the first step you have to do. Okay, unless you reach enough, you sequence sufficient amount of cells, you don't care, you just throw away those slightly lowly expressed sequence cell, and you just keep all the cell sequence to the same level. But if you don't have enough funding, you just give you one shot of the experiment, you always expect to have a long tail of this lowly sequence cells. So you have to have a way to do library size normalization to actually use some information to recover those back. Okay. By the way, we don't do the library size normalization by gene. So you see at the end, the gene average expression, the gene cross cell certainly shouldn't be the same. So some gene will express highly than other genes in different cells, right? But if you confuse with the dimension and you do the gene-wise library size normalization, you end up with something different analysis. So should we be careful about that? Okay. Again, this technology is not new. It's inherited from a traditional RNA-seq technology. So you have a raw data, and that's two samples, two columns. Okay. In this case, it's an example from a traditional RNA-seq. You have a patient, you have control, and you have 3 genes. And to do library size normalization, you see the patient has to sequence deeper, so 120, and only 60 sequence have been sequenced in control sample. So to do the library size normalization, you end up with the 60 equal number of the library size, and then you downsize actually by the factor of the patient expression. Okay, then you can compare. In traditional RNA-seq analysis, we also consider the length of gene. For example, in the C panel, this is called the RPKM normalization. We not only normalize by library size, but also divide this expression level by the length of gene. In this way, after normalization, you can compare one gene with another because this will become a density of the rates in per gene. If you have long gene, you have more rates in there. It's not fair to compare this gene's expression to the number of expression with another gene. So you divide by the length and basically the density of the rates in per gene, and then you can compare across genes. We don't do that in single cell study. The reason is the rates we get from the droplet, you know the principle, it's not the rates mapped to the whole body of the gene. So it doesn't matter how long the gene, you only capture by the poly A tail and how many. So no matter how long your gene is, you only have one poly A tail, and the rates will be found on there. That's the number of counts. So we don't care about the body of length of gene. And there are other ways to So this is called DE-seq, this R package, do the differential expression analysis. So they trade majority of genes and do normalization with these 2 genes and leave the outlier with gene 1 unchanged. Okay. So there are some special treatment because this consideration of some genes really highly expressed. And the majority of genes should be used to normalize across samples. But because the patient some abnormality in there, so you expect that some highly expressed gene is still there, and this outlier will only touch it. But that's out of the topic of this single cell. So for now, you just need to know the library size normalization. Okay. So B, yeah. The C and the D is for traditional RNA-seq analysis. Okay, how are you going to do this in MATLAB? So easy, and you sum the matrix X. Remember, the matrix is the gene by cell. So when you do the sum by default, that will sum the columns by columns. So you have the library size, and then you use X divided by library size. And the program is smart enough, it knows to divide each column by corresponding library size. Okay. If you like, you can times the factor, say 10,000. Otherwise, the normalized value is too small, maybe 0.11. And if you time 10,000, you don't change anything, and you just scaling factor to the larger reasonable level. Okay, so this technology is called global scaling normalization. There's a problem. It's blended from the real cell types, right? So you have macrophages in this case, and lymphocytes, neutrophil, macrophages normally larger, and the library size are supposed to be greater than the other cell types. But if you don't know that, you just treat the whole population mixture cell type as one identity matrix, and then you just normalize using the single factor. And then in that case, you actually push down the macrophage library size to average, and you're inflating the library size for other small cell types, right? Correspondingly, the genes like AA expressed in different ways in different proportions in different cell types will get unbiased treatment. So how can we deal with this? Any idea?
Separate out the cell types and then normalize the cell types.
Yeah, so that's a good idea. So you first group cell potential library size into bins and treat them differently. Basically, you get different global scaling factors to support the different sizes. Yeah. The problem is whether it's a chicken egg problem, whether you group them by what cutoff and into how many subclusters before doing the normalization, or you do normalization. So this is kind of a statistics problem, and the statistician, I believe they're working on those, use a multi-factor and multiple scaling factors to model, okay, to fit your data. It is a solution. So that's one of the solutions to do this kind of iteration. We try, see, okay, I assume my data have 3 categories, and then I fit each categories using different mean, different variance, and then you come back and do a loop to have a better estimation of my parameters and feed your data. At the end, you get this normalized and also cluster data. Good. This is the best solution, right? That's already you said. We are iterating this estimation and inference and also fitting into this loop at the end to get the best solution. It help us to do the normalization in a most reasonable way and also help the classification. Okay. What's the shortcoming? What's the problem with this? Why don't people do that?
Is there any problem?
You mean whether there's a guarantee you can convert this parameter estimation?
Yes, and you understand what is convert to.
I think if you choose the correct distribution model, you will eventually, and we have this called Chinese restaurant process and some statistical models. Yeah, it will guarantee you have a converged data, a solution for the parameters. This has not been widely adapted because of the efficiency of the computation. Okay, if you have 6,000 cells, there were 1,000 genes that will take much longer than you expected to converge, to finish, so people don't use it. But that so far, I believe, is one of the best solutions for this. Unfortunately, if you check the citation of this program, I don't think we have a lot of people use it. So library size normalization is still the most commonly used. Okay, before we jump into the probability distribution of data, just like I described, we need to have some kind of model to estimate the level of expression and to choose a scaling factor and in a reasonable way, whether we should consider the variance across different cells. How are we going to model it? So they all boils down to distribution of data and algorithms. Okay, let's see. I think this X theta is your model. Is the parameters for your statistical model. And you can actually use it to generate some of the data or you can just learn from the data to do this. And then you have a probability. So based on your model parameter, whether your observation is reasonable and what's the likelihood to observe this based on the assumption. So to make this more concrete, I give you an example here. Imagine you have the statistics for the men's height in European countries. And we have this generative model example game. I want you to generate the data for Italian men versus Swedish. And I don't know. Yeah, so we need to come up with some numbers. And I can invite you to come here and to write some numbers out here, okay? The procedure you are doing is doing the regenerative model. Let me switch to here. So we have Italian and the Swedish. So we have Italian here, and the Swedish here. And are you using centimeters? Okay. So I will write my first Swedish people, maybe in the centimeter, it will be 185. So Italian, who's going to give a number here? 174. That come up with another 8, another number. A number is based on your assumption is likely, the most likely number should be 190. Yeah, so that's not exactly what I want to simulate. I want to simulate around the average, right? So the average you should have 70, 168, something like that. And this here, we always have a corner people. This is certainly out there. So the game of here is, the average height of those people and the Swedish is taller, so you pop up from your idea. You're supposed to have something around 180, and I'm doing here is 170. The process here, you are doing a generative model. You use the model on your mind. This model is a distribution of normal distribution. So you have a Swedish here, having here. So that's my model on my mind. Okay, so because I know this distribution, so I can pop up those random number and supposedly to fit my distribution. If I ask you, please give me the expression in for example, please give me the CD8A gene expression in macrophage and the T cell. In this case, on your mind, you don't have this distribution, so you don't know, is this a macrophage distribution? And what's the shape of T cell distribution? This T cell. You don't have this picture for this gene's expression. So you won't be able to generate a table like that for me. You know this. You heard from the news, from your geological, whatever resource you get the information from. You don't have information for CD8A expression in macrophage T cell. You don't have this curve. So you cannot write down this for me. Okay. We come back to this Italian Swedish
拦住。
哦，是
对，有
它
点
是它可
戳出来
能没有特别正好，
对我们应该是再过来一点。
让我看看我的肾的那个单怎么还没送？
Well,
那个李维斯，
and please mute yourself. Okay, so this number you get is from statistics. So people go outside to measure people, and they have a table, and they come up with two numbers. One is the average, and then another, so the mean and the variance. So that's what we have on this chart. Okay, 1 meter 81 cent, and here 176. So you are doing statistics to get the average. That's a statistic to get the generative model's parameter. And then you can use generative model with that parameter to generate random numbers which mimic or fit the original distribution of the data. So that's why we need in single cell to build a generative model for the expression level for different genes across cells of different types. Okay. So why we need that? Because if we have this distribution, we will be able to tell whether this gene expressed abnormally high in some cell or not. Because here, for example, we know this number is really this outlier in this group. Because we know the parameters here. Same idea. So if you observe a cell, you get the CD8A expression, and you know it's in the macrophage, so supposed not to be that high, and you know something unknown in there. So that's the purpose for doing this generative model and have an estimation of parameters. No, not at all. Actually, for single cell, we don't use normal distribution because the shape is not normal distribution shape. Okay, so this diagram explains to you how this probability distribution or generated model parameter is constructed. So you start from collecting data with this discrete numbers, and then you make histogram. See, okay, I found 70% of here, 30% in this bin, and then once you have enough data points, you can make this continuous curve. Okay, so you smooth out. At the end, you get this called a probability distribution function (PDF), which is continuous form, but it's actually just a large number form for the discrete small numbers of the distribution of that histogram, right? There's no difference. Actually, in computer, you know, computer is a discrete machine, so eventually this looks like a continuous function is actually computed in the computer by the steps. The steps are still discrete steps. So there's no substantial difference between mass function or density function. It's just whether you treat this as a discontinuous or continuous distribution. Okay, so the normal distribution we use the most commonly, and for example, this means height, and because of this property, and we can use this two parameter distribution PDF. You have a mean and also the sigma. Okay, so once you have these two parameters and this curve is determined, and if you see, okay, I'm going to generate, for example, you have this curve, you want to generate a simulated data point with a mean in the middle here. And you will see, okay, I will generate this data with this value at the frequency of this height. Okay, this height indicates the frequency you want to use. If on average, the underlying curve, it has average, the underlying curve, the area equals 1, so you have to find a way to say, okay, I want to generate this by this range and how much it should be. Okay, that's your question. So what kind of model, so which distribution we should use to model the expression of a gene across cell? Same question, maybe you can also ask, what's the distribution of the gene expression of all the gene, a cell across all the genes? But I think normally we will model the genes expression across cells. So some cell will have high expression, some will have low. And from the statistics textbook, you have a multiple distribution. It has been studied before we know the properties, and you can choose from. Okay, most commonly people realize negative binomial distribution is the one we can use. Okay, you can think about this as also it's a two-parameter distribution. So you have R and P. P is the probability of the trial success. So, and R is before you get R numbers of success, how many you need to try, basically. So this is not as a normal distribution, as a straightforward, easy to understand. And let me explain it to you. So the best way to me is to look at the MATLAB function, how the MATLAB function is defined here. So NBINRD is a MATLAB function to generate random number, not follow normal distribution, not follow uniform distribution, but the number generated collectively should follow the negative binomial distribution. So that's negative binomial random number generation. Okay. So in order to generate a random number, you need to at least two parameters. One is R and another is P. And then it gives you one number. If you want to generate a matrix M by N, you can get 5, 6, and then you will generate a table, a matrix with 5, 6 random number, but collectively those random numbers should follow normal negative binomial distribution determined by R and P. Okay. So what's R and P here? So R is number of success. And the P is when you do a single trial, what's the probability to become success. So if I have a coin, I see 50% tail and head. So my P will 0.5. So I have So if I threw my coin, I think that the head is success. That R will be the number of success. I will get, so I see I will get 3 head. In total, how many trials I need? I need X times trials to get my 3 times R success by as a head, which is 50%. So that's the meaning of binomial distribution. Any questions? Yeah,
It's only for the
it's only for integer. Yes, that's discrete.
before no bracket.
Yeah, yeah. The beauty of this single cell measurement, which the number of UMIs is an integer, right? So we are dealing with integers here. So let me. So that's one example, and you have a trials for this quality control, and some product you have a defect rate 0.01. So you have 100 product, you're supposed to have 1 defective one. And how many units might this quality assurance inspector before finding 3 defective ones? So that's you found 3 defective ones, and then the defective probability 0.01, right? I will do 6 trials and see how many I need to try in order to give my 3 defective items. So the first example, I tried 496 times and I got 3 defective ones. The second trial, I did 142 times I try and I get 3. Once I have enough, I try the average of this. Okay, so I'm going to translate this into the concept, in the context, in the single cell study. So I'm going to cross over this effect into a detecting probability. So if I have a mRNA in the single cell, because of the sequencing bias on the mRNA cannot always transcribe and detect by my sequencing. So I have a very low probability to detect it. Suppose we have only 0.01 probability. If there is mRNA in the single cell, I could detect it at the end. So the probability is 0.01. Then I will see, ask, how many mRNAs, so how many mRNAs that gene should be in that cell, so that at the end, I will get the UMI count of that gene equals 3. Okay, it's in this case, because I reuse this 0.01, so this probability may be extremely low and not realistic. But I just assume we use that we have very low detection rate for that particular gene. And if I see 3 UMIs for that gene in the cell, so what's the real expression level for that gene in that cell? So I can compute that on average, you should have this. Have you get your head around? Yeah.
What is the significance of the +3?
Oh, +3 means you have 3 effective ones. So you have to have that.
But isn't that what the first 3?
Yeah, so the first 3 is actually just generate those.
That's the 3 effective items.
Yeah, that's right. Yeah, that's before you have the 3 to try. So you add 3, more accurate estimation. So somehow you can translate this and into the way you detect UMI. So you have 3 UMIs means you successfully captured that expression. And how many trials with the original cell, how many in there you actually captured? So that gave you some kind of model. Of course, this number is not real from the learning, but you get the idea. You can use this. If I gave you the UMIs, you may want to estimate 0.01. Assume 0.01 is a fix. So you know technically we have a detection rate 1% or 10%. And then you want to know what's the R on average.
I also had a question. So if you go over why was the cluster in the
Oh, and because of the definition of this is before you figure and the number of failure before you become success. You have 3 times success. You add up to the total number of trials. So you want to get 3 successful trials, and then before you have a 3 success, you have 100 failures,
Right.
and then the total number of trial actually is 3 plus 100.
But that should have been part of the function, negative binomial.
Well, you think it should be inside already. Looks like it doesn't. I copied this from the MATLAB function documentation, so I assume they have someone back them up. I just believe them. Maybe they are wrong.
So here, 496 is really 493 failures, and then
there's a
you add
three.
your 3 successes.
Yeah.
So 1 in 6 in that, or I understand what the 6 is for, you add like 6,
Oh, I just want to simulate 6 times.
right? But what is the 1? Is that just the start?
The 1 just gave you a vector. If you have a 2, then you have 2 rows of outcome. Yeah, this is just a MATLAB parameters.
Right, yeah.
How many data you want to generate, whether you want to generate a table or just a vector of data. Yeah.
One question. Do you think that any underlying insight
No, this is an open question. People, again, I leave this for statisticians. They model this and simplify. Negative binomial is better than Poisson, and certainly is much better than normal distribution because the shape is not normal at all. Okay. I think they also have the balance between the distribution. They started many years, they have experience to fit that in, and they actually, so that's again, so that's some idea about those, you model those factors in, and you have an estimation of the detecting rate, and consider all this in the factors. Okay, so once you have this, whatever you come up with a distribution to model the data, once you have that, you know how to generate the data. Even the data is not there, you can see, okay, based on my fitting of my observed data, this 0 should be 1, so I can some kind of a imputation, right? This position, although not observed, maybe 0.5. 0.5 in there. So all this model-based imputation method and normalization, so you can have this scaling factor estimated from the model, and so once you have that, the procedures you can build those in the method. Okay, so negative binomial is not the only way to use. For example, the Saver, published 2018. Internally, they use the combination of the Poisson and the gamma distribution, the they have data and they believe those are the best combinations, and they use those mixture model. And another, so this called the imputation, SC impute, another paper. I think they use non-negative, what is the model? Another mixed model, you know. Okay. So any questions before we move to another topic?
Is the connection to use the imputation concept like that?
No, it's not at all. And so whatever imputation method, you actually have some kind of a generated model underlying. We don't have a guarantee that model will be the best choice. So what we found is imputation is always doing too much to the data. And the benefit sometimes not convincing enough to let you to adapt to them. And so a lot of senior members in the field just don't use any imputation. Because once you impute your data, you transform your data, fit your understanding. For example, I transform this data, not from a real survey, but some understanding of myself. Then that will have an impact on all the downstream analysis. Have uncontrollable consequences. So, although this is a wide topic and the people still working on those, how can we impute, how can we change those values, because so many zeros, we just cannot do anything with this. So we want to use imputation to model, to fit the data, to improve the quality, but that's all based on the quality of your generated model, actually, right? Okay, descriptive and inferential statistics. So we have the position and mean, median, mode, frequency, variance, dispersions, and then on the other side we have estimation and hypothesis tests. So how can we use this? First, to know what is probability density function. Okay, PDF. On my mind, they are just a histogram, and because you make this being smaller enough that you look like a curve. And I hope everybody knows this CDF. You have PDF on the top. That's CDF is incremental, and the frequency from 0 to 1 instead of a bin, and you make them stack until you reach to 1. And similarly, so this the CDF can be expressed as PDF can be expressed with a CDF. Okay. The good thing about the CDF is if you have two distributions, so one normal curve, for example, another, you don't see the difference, but the CDF is more visually easy to detect the difference between two distributions. And I hope you know this box plot. People by standard is you have a median in the middle, and then you have a percentile. Based on the data points position, you have this box plot distribution to display your probability distribution of your data. Okay. Okay, that's an example to put PDF, CDF, and box plot side by side, up and down. And if you have a PDF in this shape, you have a CDF in that curve, and you have a box plot. And if you have a PDF with this plateau on the top, and then your CDF, you will have a long slope here, okay? And you have a wider distribution in the middle with a wider box, more data points in there. On the other hand, if you have a PDF with a peak, sharp peak, then the CDF will have a very steep increase with a jump of its frequency from just one very small incremental in the X values. You have a quick increase in the frequency of that Y-axis. The box plot will be very narrow, and the two quantiles will be splashed into the middle. If you have a bias distribution and with long tail, then you have this kind of shape. So this may be not so, yeah, so the CDF will look like that. Long tail until you reach to 1, and also by plotting bias. And if you have a bipolar distribution, you will see 2 peaks bimodal, and that's corresponding CDF. Box plot is not so sensitive to express that. Okay, when you do the inference, we want to have this distribution and have this curve, so we can compute the area under the curve and after a certain cutoff. So whether this orange area is smaller than 5%, then if you have a value located inside, it will be 5% significant. Significant means it's more unusual than expectation, away from an average. Right? Okay, I think that's all. We have a quick walkthrough of this very basic descriptive statistics. Okay. Okay, let's take 5 minutes break. Forgot to resume this. That is okay. The battery is also run out. Okay. Okay, let's see how can we calculate variance. Now remember, when you calculate the variance, we are talking about most likely the variables, the features. Translate into a single cell study that we compute the variance for genes, not for cell. So cell is a sample. So your samples is your data points. But we are talking about now the variance, which is the descriptive statistics, which is expression of that gene cross sample, cross your cell. Whether they centralized or they are more dispersed, very different between cells or homogeneous, which means expression of that gene the same across all the cells. So remember, we are talking about the variables. We're talking about the variables variance cross samples that gene expression across cells. Okay, on your screen, X is expression level, for example, for a particular gene. Gene X expressed across 6 cells have an expression level 17, 15, 15. So the average is 14. So you use every cell's expression of this gene minus its mean. So you centralize the expression. So in this case, after you centralize the data, so expression of this gene cross cell have average 0. Okay, because we put the origin in the middle of the data. To compute this, you use each data point minus its mean. So it's how far away of your expression of this gene in that cell to the average. And then you squared it and take a sum. So that's the variance. The variance of expression of this gene in the 6th cell you are considering. Okay. The variance is 33.2 here. Oh, so here we divide by and -1 is more embarrassed because this is a sum of the number of cells. So if you have a million of cells, this number will be really huge. So that's why you want to divide by the number of samples to normalize it back to when you can compare. So you have sample 100 cells and 1 million cells. Okay. Now we consider 2 genes. In a very similar way, we first normalize, centralize the gene X, okay, regarding to gene X mean. So we centralize gene Y in this way. And then we take a product of them and sum this. If you replace Y with X, you are actually compute the variance of X itself, right? So in the same way, that's covariance. Covariance is sign you have positive or negative. Now, we have an example to do this multidimensional covariance matrix, not multidimensional, but multivariables. So in this case, it's 3 variables instead of X and Y. Here, we can, if we are talking about the cells, there are 3 genes. Okay, now the column of genes, remember the features or variables. In that case, is 3 subjects, math, physics, English students, 5 students. How can you compute a covariance matrix? Covariance matrix is pairwise covariance between each pairs of these variables. So if you have 3 variables, then you end up with a 3 by 3 covariance matrix. Okay. Similarly, the first step is to do a centralization. You don't have to remember exactly how this, but that's just some matrix presentation of this centralized centralization. You can do this as what you can do here. Okay, 1 column by 1 column, and then divide by normalize it here. So once you have this centralized matrix, and also because here already normalized, so you don't have to normalize it later, then This actually is a dot product of the two matrices. Okay, so you transpose your matrix and then multiply it, and you get this covariance matrix. In the diagonal position here, the variance, because that's the variable by itself, so itself multiplied dot product by itself, its variance, and off the diagonal are the coordinates for covariance coefficient. And it should be symmetric, so MU and EM should be the same. Okay, so this is semi-defined covariance matrix. If you have multiple variables, you can do this scatter plot, and for every pairs of variables, and each blue points is a sample. So in this case, you can have a global view of all the pairwise correlation or association between variables and immediately detected. So that's one way to express this. And the covariance matrix and this correlation matrix are linked. And once you have one, you should be able to get another, because there's some kind of normalization, and then you get a correlation matrix. I will mention quantile QQ plot. Okay, so in this case, you have a distribution. Then you have data points sampled from the distribution. Okay, I see. And in one axis, you put a rank. And another, you put the value. So in that case, you will get a QQ plot. Normally, we use a QQ plot to compare two distributions. If the two distributions are the same, it doesn't matter the absolute value of those, then you have a straight line, because the quantile are matched onto each other. You don't have a quick jump in one distribution compared to another. Otherwise, you will see a distribution of those points which off the diagonal line over there. Okay, so once you have two distributions, to measure the difference between these two probability distributions, we can use so-called KL divergence. And that's a definition for KL divergence in the continuous form of this. Let me use a discrete example to illustrate. So you have a first distribution and have a histogram like this. You have three bins, 0, 1, 2, and with this distribution, you have a highest in 1 and lowest in 2. Well, another distribution you want to compare it with is uniformly distributed. So every bin have an equal number of probability. In order to compute KL divergence, you just fit all the values into that equation. But in this case, because we have a discontinued bins, so the first bin will be the probability of a P of this divided by the probability 0.01/3 and take log and multiplied by the P probability. So you do this repeat through all the bins side paired, and then you get the value of the divergence between that. And the KL divergence is not symmetric. So if you compare P to Q and Q to P, you get different values. Okay, so this is a illustrative example, but for the continuous distribution, the computationally actually you are doing the same thing. Just put this in very small, you have a sliding window, some kind of this. Okay. I want to mention this Simpson paradox. If you consider all the data points as one group of samples, you think, okay, for the two variables are positively correlated, but if you know these samples can be classified into two groups, and if you look at each individual group separately, and you see, okay, two variables, x and y axis, they actually negatively correlated. So this creates this for the statistically Simpson's paradox. So whether you should consider all your samples together, or you want to compare, for example, male, female in the two groups, and you have a negative correlation. The worst scenario is you don't know how to separate them into two groups. Then you end up with consider them as a whole. So at the end, we have the definition for probability and the likelihood. The probability is for the distribution and underline under the curve, the set out cut off what's the idea. Well, the likelihood is for the data points. So whether this the probability to see that data point for given distribution. So we use technique called maximum likelihood to find the parameters to fit your data. In that case, the data points, which are those right points in axis has been fixed. Then we can compute the likelihood for each of the data points for given distribution. And then we can change the distribution's parameters. So I compute the overall likelihood for all the data points regarding to different distribution. You have a blue curve, you have a red curve, and green curve, and then tell me which one gave you the largest distribution likelihood in total. And then you can select the best model, which could be the green curve to fit the data. Okay. If you need to know more, there are links to the YouTube. I think they give very good explanation there. Okay, so we are almost ready to do next step. Okay, we have this generative model. I hope you understand why we are doing this, how we can do this. Several commonly used distributions mentioned, and how can we compute the covariance matrix. We will do this in MATLAB. Okay, so once we have variables in the column, then how we transport and multiply and get a covariance matrix. Then we should be ready for PCA maybe next week. Okay, the homework I supposed to give you last Thursday, but the homework will be simple, is to explain this equation and its application. It's cosine, the dot product of the geometric interpretation of the dot product, and also its application. Try use your language to explain it. By the way, you did very good in the previous homework. Send me the links or give me upload your video. And one of you just upload not a video, only audio file. Because when you do this screenshot, maybe you have a separate file. One is MP4, another M4A that only have audio. Yeah, but otherwise you did a very good job. Okay, yeah, Alex.
That geometric interpretation fine, or do you want the one where I remember there was one where there was a graphical one? boxes. Triangles.
Yeah, so if you want to use my way to explain, it's okay. If you found a way to explain your understanding about this relationship, whatever make you easy to understand and remember. And then you explain some of the applications. Once I have this relationship, what I can, in which scenario can use it. And for example, projection, the redefine of the coordinated systems. Just show me the level of understanding using your own language and explain it. And then keep the record, maybe 1 or 2 semesters later you forgot and then you think, okay, I did understood it before. Okay, thank you.